Every time a cell divides, it faces a massive logistical problem. It has to perfectly copy billions of letters of genetic code in the exact right order without scrambling the instructions. Today, we are going to draw out the exact biological assembly line that makes this possible. By the end of this video, you will have a complete step-by-step whiteboard map of DNA replication. Take a look at this diagram. Notice how the original blue double helix is splitting down the middle, and a new yellow strand is locking into each half. We call this the semi-conservative model. Because each new DNA molecule conserves one of the original strands, the cell has a built-in template. This half-old, half-new structure ensures that the exact sequence of genetic information is passed on to the next generation of cells without being lost. Step 1 on our map is opening the factory floor. To read the instructions, we have to get inside the double helix as it enters the production line. Our first machine is Helicase. It drives straight through the DNA strand, severing the internal hydrogen bonds holding the genetic letters together. It pulls the helix apart into distinct top and bottom wires, creating a widening, Y-shaped opening called the replication fork. Forcing the strands into this wide fork is the only way to expose the raw chemical letters. Without this open space, the next machines on the assembly line would have nothing to read. Now that the DNA is open, we hit an architectural roadblock. The main builder enzyme that copies the code cannot start building a new strand from scratch on bare DNA. To bypass this limitation, the cell relies on an enzyme called primase. Primase drops in and lays down a short piece of RNA to act as a physical anchor point. We call this an RNA primer. This primer acts like a temporary red flag, marking the exact starting line. Without it, the entire copying process stalls before a single new letter of DNA is written. With our starting line marked, we can bring in the main engine of the assembly line, an enzyme called DNA polymerase. DNA polymerase snaps complementary pieces together, A with T, C with G. It builds only in the 5' to 3' direction, so the top leading strand grows continuously. The bottom strand runs backward, so polymerase builds it in short chunks called Okazaki fragments. This backward, segmented building process looks chaotic, but it is a necessary compromise. It ensures both halves of the DNA are copied simultaneously while obeying the directional rules of the enzyme. At this point, the primary copying is done, but the new strands are littered with temporary RNA primers and broken gaps. An enzyme called exonuclease sweeps through and strips out all the RNA primers. Then, a secondary DNA polymerase comes in to fill those newly empty holes with actual DNA bases. However, we still have a structural weakness. Even though the gaps are filled with DNA, the Okazaki fragments on the lagging strand are still physically disconnected from each other. This diagram introduces our final construction tool, ligase. Ligase slides over the gaps between the Okazaki fragments, acting as molecular glue. By sealing these fragments together, ligase transforms what was a fragile, disconnected lagging strand into a structurally secure, continuous double helix. Copying billions of letters at high speeds inevitably leads to chemical matching errors. DNA polymerase acts as its own quality control inspector. If it accidentally pairs an A with a C, it immediately reverses direction, cuts out the incorrect letter, and replaces it with the correct T. Without this immediate error correction, these mistakes would become permanent mutations, rendering the newly replicated cell non-viable. When we pull back from the microscopic machinery, we see the final result: one original DNA molecule converted into 2 perfect copies. Let's review our complete whiteboard map. First, Helicase unwinds the DNA. Second, primase primes the starting line. Third, polymerase extends the new strands. Fourth, ligase joins the fragments. And fifth, the machinery proofreads the code. This exact sequence of machines operating inside your cells right now is the unseen engine driving all physical growth, bodily repair, and genetic inheritance. Now that the cell has successfully copied its instruction manual, it has to figure out how to actually read those instructions to build proteins. We will tackle exactly how that works in our next episode on transcription.